Welcome to the webinar Perplexity is the Beginning of Knowledge, and welcome to our program IVD Ready by May 2024 with Platomics. Platomics is a Vienna-based company, and our goal is to bring safe and effective innovations to market quickly and efficiently. Our mission is to automate and digitize regulatory processes for all stakeholders in vitro diagnostics. My name is Simone Latkulik. I'm leading the customer success team for laboratories here at Platomics. My team and I support laboratories on our platform to achieve IVDR compliance for their in-house developed tests. The IVD Ready program was started to support labs and health institutions in Europe through the IVDR step by step in 1 year by 2024. Today we have invited following guest speakers. Dr. Peter Horak from National Center for Tumor Disease in Heidelberg will talk about regulatory challenges in an academic sequencing laboratory. Peter Horak is hematologist and oncologist and currently serves as deputy head at the Division of Translational Medical Oncology at NCT Heidelberg, where he is involved in the establishment of the Interdisciplinary Laboratory for Precision Medicine at the National Center for Tumor Diseases Heidelberg. He studied at the University of Vienna and did his postdoc research at Harvard Medical School. Erich Zeisel will talk about regulatory challenges will talk about terminology, terms, and definition of standards. Erich Zeisel is head of production at Austrian Standards since almost 20 years. In his role, he is mainly responsible for technical content preparation, quality assurance, production organization, and policy creation. Marisa Lien will talk about knowledge transfer from a practical kind of view. Marisa Lien has years of experience in medical device and IVD approvals. She's not only a product specialist for IVD medical devices, but also an independent consultant and auditor. As a consultant, she cooperates, for instance, with Biomed, but also with TÜV Austria for training and lectures. Last but not least, Andreas Oberleitner will give a demonstration and walk through the PlatoX IVD Assistant. He will present the Platomics glossary, IVDR glossary, and also the classification wizard on our platform. Andreas Oberleitner is head of QM and regulatory affairs at Platomics. He has more than 20 years of experience in this field. He is especially an expert in the domain of conformity assessment. During the webinar, you can always ask questions in the Q&A section, and we will try to answer them by the end of the webinar, if we have enough time. Now, the IVDR came into force, as you know, last year on May 26 in 2022, with the first set of requirements for in-house developed diagnostic tests. Laboratories and health institutions in Europe now have to fulfill the Annex 1 of Article 5.5 of the IVDR. The next set of requirements will become effective in May 2024. The IVD Ready program by Platomics was started to educate and enable laboratories to achieve IVDR compliance by 2024. During this program, we offer each month a webinar focusing on a specific theme that will be held as shown here on this slide. But what makes this program really unique is the fact that we not only educate, but also enable all participating laboratories to create complete in-house IVD documentation via our digital platform. To actively participate in the IVD Ready program, it's really essential to use our software tool, the IVD Assistant. Attending the program and using the IVD Assistant are both free of charge exclusively for health institutions and diagnostic labs. Why is this so important? We release templates and wizards in the IVD Assistant on our platform during the program on different topics that are necessary to achieve the IVDR compliance of your in-house tests actually by the end of 2024. Now we will start with today's topic. It's all about knowledge and knowledge base, entitled Perplexity is the Beginning of Knowledge. I will hand over now to our guest speaker, Peter Horak. Peter will talk about his lab and regulatory challenges from a lab perspective. His lab recently got accreditation, and I'm very happy to announce his contribution to our webinar today, Regulatory Challenges in an Academic Sequencing Laboratory. So please, Peter.
Thank you, Simone. Thank you for this nice introduction. Let me share my screen. So I hope you can see the screen now. So first, I'm a board-certified and trained hematologist and oncologist, so not a laboratory leader or per se, but I work at the German Cancer Research Center and at the National Center for Tumor Diseases. And in my position, I co-lead a large sequencing program, which I will explain to you in a couple of minutes, or bring closer to you. And then in the second part of my talk, I would like to focus on what challenges arise through the IVDR for our lab or for our institution in terms of performing this sequencing procedure. So at the NCT, we have two major possibilities to do molecular diagnostics. First, through the Department of Pathology of the University Hospital of Heidelberg, where there's the opportunity to perform gene panel testing, whole exome sequencing, and it's all standardized or accredited to the ISO 17020. But I won't talk about that part of our sequencing platform, so I will talk about more specialized programs which have arisen through an academic collaboration with the German Cancer Research Center, and among those, I will focus on the master program. So the master program is a cross-entity prospective sequencing study focusing on rare cancers, advanced disease, and is currently running since 2013. We have collaborators across the whole of Germany and even abroad. We have more than 100 centers and partners who send us patients. Among those, there are all National Center for Tumor Disease, all DKTK and CCC network sites. So we have the necessity for this because of our focus on rare diseases, but we have to cover the whole spectrum from private practices to the university hospitals. More important, we follow standardized workflow for a precision oncology program involving all steps from patient registration, sampling of blood and tissues, molecular profiling, clinical curation and data interpretation. We provide a molecular tumor board currently four times a week, and we give treatment recommendations which are then implemented in clinical trials or within an off-label treatment. And that's the important scope here, because for doing a diagnostic procedure for clinical trials, our procedure has to be accredited and IVDR conform. So since 2013, we started with the whole exome and transcriptome sequencing. We added germline evaluation with our colleagues from the NCT in Dresden in 2015. And since 2016, the majority of our patients has the whole genome sequencing. So these are the diagnostic procedures which we need to have accredited and IVDR ready. Until now, we have enrolled over almost 6,000 patients and analyzed over 4,000 within molecular tumor board. This is only a small fraction of what the German Cancer Research Center has been doing in the last 10 years. So at the core sequencing facility, there have been over 40,000 exome and genome sequenced and over 30,000 transcriptomes, mostly though without any clinical association or without any clinical impact. So that's what led us to the establishment of laboratory, which has this nice short name, Interdisciplinary Laboratory for Precision Medicine at NCT Heidelberg and Center for Rare Diseases in Heidelberg, and even a nicer acronym, NCT ZSE-ILP. Nevertheless, it incorporates laboratories or divisions from several institutions. Among those is the sample processing lab at the KFC, which is already ISO 17025 accredited. The same is true for the next generation sequencing core facility, also accredited for the ISO 17025. And the whole sequencing procedure will be accredited to the ISO 15189 as a medical laboratory, hopefully next year. We decided to follow the norm ISO 15189, in contrast to our colleagues from the pathology departments who have their own norm while performing the same test. Nevertheless, this involves all steps from administration of the samples, sample preparation, sequencing, automated bioinformatics, and translational bioinformatics. So we have to separate those two, as some of them have to be medically supervised, and of course then the genomic part. Not part of the accreditation or of the IVDR are the final discussion or the clinical decision-making in the molecular tumor board and the final report. So these are exempt from most of the norms as far as I know. And first of all, we have to ask the question, so what do we need to be IVDR conform? So genetic testing, whole genome, whole exome, transcriptome sequencing are all within the scope of the IVDR, and we use them in-house, so they are marked as LDTs, although IVDR never uses this term per se. But it allows the in-house use of lab-developed test devices, and provided there is no marketed device that fulfills patient needs, and there's currently none, as far as I know, providing whole genome, whole transcriptome sequencing at this scale. We have to adhere to Article 5, Paragraph 5 and Annex 1 of the IVDR, but first of all, we have to be accredited, and IVDR provides several options, and one of them is ISO 15189, which we chose to follow. Our colleagues from the pathology department published recently a nice discussion and of the IVDR in a pathology journal in Germany. It's in English, so it's accessible to the whole world. But it's nice to see what their approach is to the IVDR. It also involves some discussion of how the ISO standards which they use, so the 1720 norm, are compatible with the IVDR and what is needed to fulfill all the requirements. And currently, we actually don't have a solution, although we are working on one together with Platomics. But we are happy, so we are at least comfortable with the use of ISO 15189, as some of the IVDR requirements are already met within the ISO 59. So these are the chapters of the ISO 59 which are already kind of part of the IVDR or could be substituted for the IVDR. So we can conclude that the requirements of the IVDR align well with the ISO 15189 framework, and it's a good alternative. So the ISO norm, which we have chosen to follow, to national provisions or the ISO 7020 or 7025. So our current solution, nevertheless, is to do it all in an Excel format, which of course we are aware is probably not the best one and not the best one for the future, as we may have a need, you might have noticed in the following slides, to extend the scope to other methodologies or to other methods, as proteomics or epigenomics, which are currently not in scope of the laboratory. but to do that more efficiently, we will need some help. And as a discussion starter, maybe for our discussion afterwards, and final slide I would like to show you are some statements from stakeholders. Some are from the literature, some are from my personal experiences with people when we discuss IVDR and when we discuss what might be necessary to do and how this might affect our workflows. So first of all, there are several statements which might be counterintuitive, but nevertheless are helpful to understand how people think and how we think also about the IVDR. So for example, I pose it as a question here: do modified CE IVDs, which are used on a regular basis in pathology labs, which are evaluated and documented according to regular ISO standards, as LDTs within the IVDR, might there be a problem with the lack of commercially available alternatives to LDTs, which means that we run out of tests at some point? Is there a lack of sufficient professional awareness and involvement? And I can contribute to that from my own personal experiences, is that until recently, so I would say 2 years ago, no one was really talking about IVDR, at least not in my sphere. And there might be some difficulties in implementing IVDR also at the national or supranational EU level, because there's a provision to provide operational expert panels, and there is currently one, as far as I know, for the IVDR. And I'm not really sure how operational it is, and it has to provide expertise to the EU and to the notified bodies for all tests and which are covered by the IVDR. And of course, the increased demand and shortage of notified bodies itself might pose great challenges for many economic actors. There are currently 3 in Germany, 1 in Austria, so we might be well covered for the future. Nevertheless, other countries do not have even one, and there might be even still more demand than there are notified bodies left. And of course, this statement is taken from the publication I just showed you, is that some of the Article 5.5 provisions are equivocal at best, so that there's might be some space to shape some of the statements or interpretations of the Article 5.5 to be shaped by stakeholders themselves. And I think that's what we also might talk about. Nevertheless, that was a short introduction to our laboratory and to how we work at the NCT in Heidelberg. I hope you have now some overview of our programs, and I would welcome any questions at the end of our presentations. Thank you.
Thank you, Peter, for this very nice insight into your laboratory and your work, and also your view when it comes to the accreditation where you stand on the IVDR. Okay, so we are trying to make the webinar as interactive as possible, and therefore we always ask a question to the audience, and you can answer the question within a poll anonymously if you want, and we are going to show the results then later after on. Question is, accreditation ISO 15189. What is your current status? Not yet started, implementation ongoing, certificate received, or not applicable? Other standards may be implemented. And now I will hand over to our expert that will talk about termination, also terms and standards, Erich Zeisel, about terminology, terms and definitions of standards.
Thank you very much, Simone. I would like to share my screen with you. I would like to talk a little bit about the terminology and standardization terms and definition within standards. And that raises the first question: is terminology for standardization important? And the answer is, of course, yes. And there are some reasons for that. And I would like to start with the dry facts at first. So we all know there are a lot of very important standards around this world, and Austrian Standard has made it its mission to incorporate all these important standards into our database, and in most of the cases on a weekly basis. So we have all the European standards, the EN documents, all the international standards, the ISO documents, which we'll hear about that a little later, and the vast array of standards and technical regulations from other national standards bodies like Germany, the DIN documents, England, the BSI documents, and yeah, not to mention all the exotic documents from all over the world. So question raises, what does all these, or at least most of the standards have in common? Well, it's a consistent structure and format, and thus a unified, or let's say a standardized terminology. So why is that so? Yeah, well, because we are a part of the standardization world, and this is our main goal. Yeah, we have 24,000 documents in our catalog, and that ensures safety for the consumers, for the economy, for science, and for the state. So nearly all of these documents contain terms and definitions. And according to our latest calculation, there were approximately 800,000 terms, and as a consequence, we have about 800,000 definitions as well. And to main clarity here is of the utmost importance to standardize these terms in form and structure. So important aids in this regard are term standards, internal regulation, and guidelines that include specific specifications and how the architecture of these terms should appear. So it would be beyond the scope to delve in all these regulations, but it's important to understand that this uniform format is to be applied across the entire portfolio. So no matter whether you're reading an Austrian, and German, an Italian, English, or an international standard, you will discover the following in all of them. There are terms and there are definitions, then they're always located in clause 3, and the terms are not consistently numbered, are consistently numbered. The terms always have the same structure, they just as their definitions do. So notes with additional informations are constantly formatted, and synonyms are positioned in the same place. Figures are placed before references to scopes, and so on and so forth. So from a linguistically point of view, that means that the terms and definition clause is a normative element. It's define in which the listed terms shall be interpreted, and they never contain requirements, and but rather describe the nature of an object or a subject. And from a technical point of view, that means that yeah, we can ask why it is this have this strict structural framework, and why is it so crucial to have that? And this is because with such a high amount of terms, it's no longer feasible to manually process, exchange, and further manipulate this data. So for this reason, we prepare the terms and definitions for XML format and perform semantic tagging on them. And this means we instruct the application on what constitutes a definition, what a synonym, and how notes should appear, and so on and so forth. So as a result, we can input and manage all these 800,000 terms in databases and search engine as well. As a next step, ICS classification sort all of the terms to their specific topics And so we can provide specific fields with the right content, and even more, we can identify the disparities within the terms easily. So here I brought you a short example of a term and a definition, and you can see easily that you have different definitions here for the same term. And on one hand, you have the owner and possessor, which raises the question, if you possess a thing, does that mean you are the owner as well? And in one definition, you have the storage and transportation. In the other, it is missing. So does that mean it's not relevant to the meaning, or our legal provisions go with it? And finally, you have you can see that the term waste also appears in the definition, which is absolutely a no-go situation, because you have to define waste in other words, so everybody can understand what is going on here. So apart from misuse and interpretations, it's easily understandable that such differences can also lead to legal consequences. Therefore, we have a database, and this database features a learning tool. We have it is multilingual. We have ICS search. We have a document search. We can import Word, scan, we have scan import, XML import, and we all manage that over an API for integration into other applications and various user levels. Our target groups are end users of standards, and the terminologists, of course, translators, committee managers, members, content developers for software, and other commercial third parties. So as a result, the most important fact first, it is really necessary to have consistency and clarity into bring consistency and clarity in your terminology. So we have to ensure that the specific terms and definitions are used constantly throughout thousands of standard products, making it easier for all the stakeholders to understand and apply the documents correctly. Why is that so? Because it refuses confusion and ambiguity. So that means we avoid a lot of trouble at the end of the day. It strengthens the interoperability. So the content of standard is the final product of collaborative work that involves different organizations, different industries, and different countries. So a common terminology ensures all parties involved have to share understanding of key concepts, which is crucial for effective communication, collaboration, and interoperability. We have legal and regulatory compliance aspects. So in many cases, standards and even other technical content have legal and regulatory implications, often part of a contract or incorporated into law, national or European, the harmonized standards. And as I mentioned earlier, a well-defined terminology helps to precise definitions, reducing the risk of misinterpretation and non-compliance. One big issue is the global adoption. We all know we live in a global world, and many standards have international relevance and are adopted by various countries. So a terminology database and even glossaries help to ensure that the language used in the standard is appropriate and understandable across different cultures and languages. And it facilitates widespread adoption and implementation. It is useful and necessary for effective or efficient translation process, So for national, European, international standards, accurate translation is a necessary part, and glossaries and term bases aid in maintaining consistent translation across languages, and they are preventing misinterpretations that could arise from insistent terminology usage. Term bases are mighty tools for knowledge preservation. So what does that mean? Standards always reflect the accumulated knowledge and best practices of a specific field of expertise. Term bases and glossaries act as a repository of specialized vocabulary, and they ensure that the terminology remains consistent and can be passed on to reviewed documents, new revision, amendment, correction notice, whatsoever. And the last 2 points, we reduce cost and errors. You can save a lot of time, believe me, money and resources by setting up terminology correctly. So that means unclear, confusion, and inconsistent terminology leads to errors, disparities, misunderstanding, rework, and increasing costs. So therefore, centralized terminology, on the other hand, helps to prevent errors, support efficient communication, and saves a lot of time and resources, believe me that. Last point is the revision updates. Standards and their encompassing terminology are systematically reviewed for currency, and term-based facilitates these revisions by providing a structured framework for incorporate new terms and definitions while maintaining existing ones. Well, thank you very much. And I hope there are some questions at the end. Thank you.
Thank you very much, Erich, for your contribution and a very interesting talk on terminology, terms and standards. We will now go on and we will share the results of our poll with you. Just give me a second to share my screen. Okay, this would be the results of the poll, accreditation ISO 158189. What is your current status? 10% answered not yet started. 38% answered implementation ongoing. 35% answered certificate received. And 17% answered not applicable because maybe there are other standards implemented. So thank you for your contribution to the poll here. And now I would like to hand over to Marie Saline. She will give a talk from her practical view, about knowledge transfer.
Yeah, hello. So I'll begin immediately by sharing my screen. I think it will be easier. So like this, so everybody has the view on my screen already. So yes, perplexity is the beginning of knowledge. And what is knowledge? This is the first question that we have. Definitely, I'm accompanying as a consultant medical labs on the travel to IVDR know-how. It means that I have a passion for everything what is IVDR. I have a passion what is a routine lab in the laboratory. And I would like to bring to you today a little bit of insight, what I made in the last time since the publication of the IVDR. We all know to live means to learn, We cannot make our life a personal or professional life without to learn. And definitely the IVDR was immense impact on learning, yeah, in the practical life of the laboratory. I mean, in university, in research, yeah, we are loving to expand our know-how. We are permanently searching to have the intellectual interest to learn, to get more information. In the routine, yeah, of the diagnostic in the laboratory, yeah, we have definitely a growing volume of know-how. It means that this growing volume is sometimes simply overwhelming in the routine of a laboratory because it's demand an increase of the resource. Sometimes it creates in the daily life disbalance in the different workflow. So case-by-case activity, organization-based can be developed to make know-how transfer more logical. Yeah, but the laboratory are definitely facing challenge in the routine that are having an impact on their routine primary target to make diagnostic. So one of the first things that I'm asking when I try to when I'm accompanying laboratory to their travel to IVDR is to ask, do you speak regulatory? Because this is one of the first contacts that you have with a new know-how is sometimes a new wording. So you have in the routine, the laboratory medical scientific wording. This is clear. Yeah, it was a long education for most of the laboratory person, from the doctor who is validating, for the biomedical analysis that are working in this field. So scientific wording is something that is quite clear to them. But then you clash this to a quality management wording that is sometimes very different. Yeah, so you have really a clash, and it feels sometimes a little bit like the House of Craziness in the Asterix comics, yeah, where you have different wording giving another feedback of what is requested. A good example is verification and validation. Yeah, when I explain to a clinical team that they have to list first what they would like to reach, it means the input into the development, or that they have to make requirements that are not purely clinical. I have, I don't know, 10, 15 pairs of eyes looking at me like if I would speak Chinese. Yeah, the one of the first steps that is in the practical view necessary is to clarify what is my wording. ALAP, ASAP, LTD, PMS, PMPF, all those words, yeah, are some things that are not really part of medical education, that are not really part of training that are done for a diagnostic. So in this case, yeah, we have also to summarize what is the know-how. We have a know-how that is coming from the clinical literature. Yeah, we have a know-how for a new device, for employee management, for incorporation plan. All of this is a know-how that is inside the organization, but also have a correspondency in the IVDR, competencies, or further education. It means that many laboratories have a certification following ISO 9001. This is quite usual. Yeah? So to talk about data know-how management, this is something that is quite well implemented in the 9001. What is a little bit more funny is when I ask, okay, how are you doing it concretely? And then the answer that I receive is I will say in 60%, 70% of the cases to say, oh, we have a server, or we have a library. So it means that people know where they possibly will be able to find the information, but it's good at least. Yeah. But the question is how far they can explore this know-how, how far I can make an adequacy between the know-how that is already in the laboratory and the know-how that is requested from the IVDR. For example, so the source of know-how, yeah, the IVDR is growing. Yeah, the request that we listened before in the previous speaker about the ISO 15189. Yeah, this is an accreditation standard. So it's not a simple certification like 9001. This is an accreditation. So it means that the management of the know-how requested is much more bigger. And this is something that you cannot manage only with a server link, with a library, with a place on the server. And when I work with a laboratory to implement this 15189, yeah, many times we are facing challenges that are not only that the people have to learn it, but simply practical. Do we need a new server? Do we need a room? Do we need new meeting? Do we know? This is all this around environment that is growing. So we have a maintenance of the know-how and we have an exploration of the know-how. It means that both parts, yeah, need to be explored vertically and horizontally inside of the organization. Knowledge is definitely a critical resource, yeah, in time of digitalization. Yeah, I suppose this is in full Europe that we have this question with personnel, yeah, and employee in the laboratory will maybe change the employers more often. And so the knowledge might be lost, or we have a big part going to retirement. So it means that information, yeah, is much more important as a few years ago. Yeah, information also is changing faster. Through the digitalization, we have a higher turnaround inside of the data. Yeah. So as a result, yeah, when searching for information, yeah, the teams, the personnel, yeah, they can often find incorrect answer or find statements that are not fitting together. I had this in a laboratory in Vienna, yeah, where definitely I had one laboratory, two departments, and they had different information. So their process was simply not fitting together because they were picking the data, the document on another place that it was supposed to. So searching for the right information, yeah, is costing a lot of time and money. Yeah. So when we come back to the IVDR, yeah, knowledge in the IVDR is the ability to share, yeah, but also to learn a new language inside the team and for the future audits. When we want to avoid that we have a negative effect with too much information, though the personnel in the laboratory has already a lot of tasks. So the idea is really to present the data and the know-how on a way that you have access to the correct information and the one you need. Yeah. So when we talk about the IVDR, we have many times in the text of the IVDR the word the latest scientific knowledge. I will say when I'm asking a laboratory personnel, a team, a clinical institution, what is a scientific know-how? They will all know about it because this is exactly the core of the activity, clinical know-how. Huh? So this is a well-known process, definitely. But with the IVDR, we got added requirements like the ISO 15189. We have added requirements in creation of new document, of new type of validation. So it means that it's the pressure coming from the IVDR and complementary work to make a dedicated wording for my organization. So the challenge that we have when we are working together with laboratories, yeah, is to say, okay, what we are talking about. We are talking on this word coming from the IVDR, so the regulatory side that I bring to the discussion, yeah. But on the other side, they have also to make clear for the team, what does it mean? What is the process exactly that will fit to it? A good example is simply performance evaluation. When you speak performance evaluation inside of the laboratory, so we say, oh, yes, we validate the process. Everything is functioning fine. And then you have to say, oh, wait, the IVDR is requesting also that you describe your inputs, that you describe the intended use of your product. And the term product itself, sometimes it's very confusing. So the knowledge creation for the IVDR, for me, it's definitely something that is not a pure goal, because when you're simply saying, oh, I want to reach this one, you place the team in the laboratory in front of a challenge that seems far too high. So the process to find this know-how, this knowledge, is definitely a step-by-step. We have small goals. First is a glossary, what we understand between all those words. Second, what are our process? Third, what is our product? Fourth, where we will document this one. So this is really very critical steps to resolve before going in the project of the IVDR. When we talk about know-how sharing, further the project management, we see also a complete structure inside of the organization. So we are talking about organization culture, about know-how culture. So in the 9001, you have a know-how management responsible person. But here we have to go deeper. And I have example of what I mean with a know-how culture. I have laboratories where I have really created since 2017 a special quality management circle. And it's not only the chief of the laboratory and the quality managers sitting together and making their own soup. No, they're really proactively informing, discussing with their team. I think this is something that is quite normal in clinical point of view. All the laboratory almost have a labo jour fixe, a meeting where they are discussing about laboratory questions, clinical questions. But to have a topic definitely on the quality management jour fixe, not reserved for certain people, is very important. When you are applying for 15189, this is also to make a quality management circle. It's very interesting because you get two targets with one action. You have the further education of your team, and you have the know-how sharing. So we want to use knowledge efficiently. Something that is sophisticated enough, but according to the user. I don't want to sound too mean, but definitely when you are in the laboratory, you have different personalities, and for a member of an organization to offer his know-how proactively, this is possible only if the complete system functionality, but also the organizational culture are the decisive factors, So this culture to share information, to have a better comparison, to have a sharing of the practice, need structure and a simple access. If you ask a biomedical analyst in the laboratory chain, in the routine, most of the answers that I get, tell me what I should do. Okay. This is good, I will tell you, but we have also to exchange on this know-how. And the IVDR has this really through the different level of the laboratory requirement to understand what is the performance evaluation, for example. When we talk about access, simple access to a data transfer, to know-how transfer, this is on an individual base, supported by a structure, and after you have to evaluate it, know-how offered is good, but know-how where we could check if the retrieval of the content is possible is also a necessity. So it means that to have this culture of know-how inside of the laboratory that will support your process in the IVDR definitely need implementing guidelines. So coming to my last, one of my last slides, when we're talking about regulatory requirements in the laboratory, we are talking also about a transfer of a totally new field. With the ISO 9001, I see many times in the laboratory, the responsible person for quality management, and when I ask, how did you come to the job? And I received the reply, oh, I was designed it. to make this one. So it means the dedication of the role for the regulatory requirement in the IVDR is critical. It is more than only a decision. It is a complete education, what I'm supposed to do. Yeah. The primary role of laboratory are definitely patient diagnostic. So I never saw a lab technician sitting and doing nothing. So the will to apply for regulatory requirement will always look like much more work. And when I show you these small memes with the phone, yeah, it's because it's something that I see many times. When I ask, okay, where did you document this one? Or how do you send this to the responsible? Oh, it's not once that I receive the phone, says, oh, I sent her a message. This is definitely an information that we are losing. That's why, for me, the critical point on regulatory requirement, know-how transfer is based on the new approach in the laboratory. And here, this is definitely my practical view, yeah? But I have the opinion that the laboratory, they know the regulatory, they know the clinical needs. We are doing massive training. There's a lot of academy for quality management. But what the laboratory needs, yeah, is to be supported in the how, concretely, not theory. Tell me how to doing it. And here, then we will have a knowledge transfer based on a question of structure and resource. This is, for me, the main point that I took back from my travel with the laboratory team on the way to the IVDR, because definitely one does not simply walk into the IVDR. Thank you very much for your time. And if you have questions, I will be glad to reply them.
Thank you, Marie, for your contribution and very interesting talk. I will now hand over to Andreas. He will give a demonstration and will show us our IVDR glossary and the classification wizard. Andreas?
Yeah, thank you very much. Good afternoon from my side. I would like to take on this figurative comparison of just imagine that we are navigating through the dense forest without a map or a guide, and the path is somehow winding, and the soil is unfamiliar, and so on. And this is how the regulatory landscape of the IVDR can feel for many of us in the laboratory world sometimes. Because the IVDR, as we heard several times, is really filled with intricate terms, definitions, classifications, and so on. But it's not just about understanding the single terms, but also grasping how these terms interconnect with each another. And that's also the challenge. How do we fit those complex regulatory pieces together, and how do we ensure that we are not just compliant on paper, but really also in practice? And I think that the answer definitely lies in education, familiarity with all of these terms. And by delving deep into this IVDR terminology, by understanding these small nuances and so on, we can really empower ourselves. And I think that's really needed. So we need to walk through that IVDR forest. And for that, we would like to equip ourselves with the knowledge. And for this purpose, we need to have the tools we need. And to support that journey, we would like to introduce our first small tool, which we now built into the assistant very recently, which is the glossary. And for this purpose, I would like to share my screen with you. I have only one small slide prepared for that, just to give you the better overview. We already learned that there might be a big confusion on so many complex terms, and the reason is because we have different sources, a lot of research is needed. Where to start from is sometimes not really clear. Every single word might count and might be important. There might be a lot of misinterpretations in those terms. And last but not least, not to forget that those changes which might appear in the definitions could also be problematic. Our technical solution for you right now is to implement a knowledge base into the IVD Assistant. And we are starting with the small regulatory affairs glossary. which already contains right now approximately 600 terms from different legislations and also standards. So the IVDR, of course, is in the center, but also MDCG documents, different laws, and similar sources will be part of this glossary. And this glossary will be continuously growing. And one additional benefit we believe is that we are really putting the source as a hyperlink into the entries themselves. You will see that a bit later how that works. So you can easily access those original resources as well. The ISO standards, the definitions which appear in our tool are with the kind permission of the Austrian Standards Institute. This is how it looks like. We will see that a bit later. Another tip I would give to you in that connection is that we can use this resource very well also as a training resource directly in the tool to get familiar with all of these terms. Okay, let's switch immediately to the tool itself. So anyone who was already participating in the prior webinars knows already that we had already created a lot of demo projects within our environment, and I will reuse the old demo project here, which was this real-time PCR workflow for HIV. And as you can see, some of the things have been already pre-filled in the former webinars. I will not go into much detail here. We have already pre-filled general information. We used the intended purpose wizard, and based on this, we could already create more or less 100% automated the intended purpose documentation and also documentation on the qualification as a medical device. So the next step for us would be the risk classification. I will come to that a little bit later. Before that, I would like to show you this additional glossary functionality. You can find it in the help menu, and if you click on the term glossary, it would open a second tab, and you can easily use those two tabs in parallel. And this is just the beginning. So what you can see here is the collection of around 600 terms, including all of these definitions here. I will not go too much into detail. I think it's very clear that those definitions are taken from the official sources and can be read up during working on the system. In future releases, we will definitely connect it also context-based, which means that you will get even a better user experience also in future releases. But for now, I think it's very straightforward and quite easy to navigate through the different letters with the abbreviations, and you can find out about the different terms. If we, for example, have a look into the term calibration, you can see this term calibration's definition comes from a standard from the EN ISO 17511. And if you click now on this one, then you will get automatically redirected to a page that gives you more information on this standard. And in that case, you can even do a preview. I need to mention that this is not part of our own application. This is already part, as you can see, from the Austrian Standards Institute, because we are redirecting directly to the information page on this standard. And if you're interested, you can even open a preview on the standard, which is made available by the ASI. Okay, this is the part with the glossary. Due to the time, I think I do not need to explain a little bit more. You can explore on that on your own. And I would like to get back to the classification topic, because one of the terms we found definitely is the risk classification. Yeah, we are now in the step of or in the need of classifying our device or our workflow, and for that we need to know a few things. So in general, this risk classification is kind of a framework to categorize devices within the IVDR according to their risk level, and we know that the goal is to ensure a certain patient safety and also to derive measures which are fully applicable to different levels of risks. In the old system, we know that there was only this list system for manufacturers, which was has not been extended during the past 20 years. And now with the new system, we have these 4 risk classes, which follow a certain decision tree. From A to D, A is lowest risk, D is highest risk. You do not need to know all the details yet, but it's important to know that for manufacturers, the risk classification has a big impact on the assessment within the conformity procedure. So higher risk classes require definitely more rigorous assessment and even the involvement of notified bodies. Yes, I will jump over these points here because they are more for the manufacturers. But now the question arises, what does it mean for laboratories? Because, you know, the Article 47 and also this Annex 8 on classification originally is not applicable to in-house devices a priori. So there is no explicit need for that. But it's important to recognize that the Article 5.5, which fully applies to in-house devices, has certain requirements depending on the class of IVDs. So as you can see, for example, in this point 5, the grade of documentation or the extent of documentation is dependent on the risk class, and therefore there's an implicit requirement that you classify your devices. And a second reason, it is generally acknowledged state of the art that a certain risk determination and also derivation of adequate measures is based on the risk of a device, and therefore such a classification would be very helpful for yourself to determine the risk of your device and also being able to derive the right measures. So state of the art means also, for example, this IMDRF document, which schematically indicates that with a lower risk, you have lower requirements, and with a higher risk level, you have higher requirements, very generally speaking here. Okay, and how do we tackle that within the tool? We implemented a risk classification module, which is literally a wizard helping you in determining your risk classification and at the same time using that information to pre-fill and pre-populate more or less fully automated the documentation for that purpose. So on the one hand, we have that wizard helping you in collecting the information. It's again a kind of a questionnaire. You will see that in a minute. And the second part of this release here is that we provide you with a document template on classification, which, as you might already know regarding our approach, will be pre-filled and automated for you. So this is how it would look like. But enough of words. I would again jump to the application. Pick up our real-time PCR workflow again. And as I already explained, these things here have already been pre-filled. And now we can enter the risk classification wizard, and it looks again very familiar for us. It's a questionnaire asking you several questions, and I will quickly walk you through those questions. So the first question is, will it be combined with different products outside of the product under scope itself? I would say no for now. Second question, is it an accessory by definition? This is also no for this case. And if we keep in mind that we have an HIV test, then potentially we think of samples taken from very special patient groups, including pregnant and newborns. I will now include this depending on my intended purpose, for the example. Then I switch over to the next page, and there it comes with more questions. So first of all, it asks me if the tool is intended to assess the suitability of donor blood with respect to transmissible agents. I would say yes. So our test is indeed intended to do so. The next question is, can we detect the presence or the exposure to transmissible agents that cause a life-threatening disease? Again, I think this is clear that this is yes. In the next question, we will find out if We have things which are influencing the patient management options with potential life threats. Again, this is yes, and so on. So next one asks you about immunological compatibility of blood components. Would say this is no in that case. And then we have some more detailed questions. Is it intended to be used for detecting the presence or exposure of a sexually transmitted agent? In our case, yes. Second one is cerebrospinal fluid, the blood which does not have higher suspected high risk. I would say no. The next one, infectious agent to the individual fetus or embryo being tested. I would say yes for my case here. Prenatal screening, would say this is no. And then we have also determining infective disease status or immune status, where the risk is that an erroneous result would lead to patient management decision resulting in a life-threatening situation. Again, I think this is applicable for our result, for our example here. Okay, the rest I would like to leave empty for now and continue and say, okay, this HIV detection PCR is intended for self-testing? No, because it's for professional users only. And do we deal with any of these following subgroups? So those are more the general devices like general instruments or general laboratory use or even specimen receptacles. But as this is not the case for the overall workflow, I would say none for now. And do we have a quantitative or qualitatively assigned value? Again, in our case, this is yes. Okay, and that's how we ask you regarding questions to determine the classification. I will now save and create the document. You see that the saving was successful, and we are now able to access the assigned relevant document, which is the classification documentation. I will quickly enter the document itself, and as you can see, there is again our document editor, which is already pre-filled wherever it was possible. First with the product identification, and also the purpose, which was derived from the old project we already showed to you. And then we continue with the classification checklist. And as you can see, this list is already completely pre-filled. So you do not have to care about thinking of if the implementing rules are applicable and if they are considered. As you can see, this is already pre-filled. And the classification rules, those 7 rules which are documented in the Annex 8 of the IVDR, they are also already processed and handled. So rule 1, rule 2, and so on. And for each of the sub-elements of these rules, you find the evaluation, yes or no, if that applies or not, including a predefined text element for justification. So scrolling through the whole document, I will just bring you down to the final result. It turned out that our device seems to be obviously a risk class D, which is quite obvious for a test which handles HIV, and therefore this document determines that first of all the risk class itself is a class D, but it also gives you the reason because the rule 1 indent 1 is applicable and so on. So you have everything automatically pre-filled by the IVD assistant, and you can immediately export the document if you want to and store it with your documentation. So there is the PDF. Again, you see it's pre-filled with all of the parameters and values, and yeah, that's it. Okay, that was it also with my presentation. If you have any further questions, I'm very happy to answer them. Thank you.
Thank you very much, Andreas, for your contribution and the demonstration of the glossary and the classification wizard. So, yeah. As I said in the beginning, to fully participate in the program, access to the IVD assistant is essential. You can contact us via email, or you can also register directly on our website. to the IBD assistant, and then you will receive your user credentials via email within 5 business days. Also, if you would like to register for the whole webinar series, you can do that easily via email, just sending an email to info@protomics.com. With this, I want to express my sincere thanks to all participants who have joined us in our program IBD Ready by 2024 from around Europe so far. Labs can still join us, and so you can invite your colleagues to and share the program. A big thank to Peter Horak, Erich Zeisel, Marie Salin, and of course Andreas for their contribution today and the demonstration and giving the talks. And last but not least, a big thank to our team, especially our software developers and the regulatory team. They work with high speed as always. So thank you too for making this program even possible. Thank you. With this, I wish you a wonderful afternoon on behalf of the Protomics team.